Hello, welcome to my virtual poster. I'm Dimitra Sarantopoulou from National Institute on Aging, and I will talk to you about our trip to integrated profiling of transcriptome and methylome of human immune cells in aging. Our objective is to identify aging transcriptomic patterns in immune cells, their co-expression and predictive expression profile, while taking into account the epigenetic regulation of age-associated gene expression. We use bulk RNA-seq and DNA methylation data from the Gestalt study, and specifically 13 cell types from 75 healthy individuals, 20 to 90 years old. Specifically, we use PBMCs, B cells, naive and memory, T cells, naive and memory, monocytes, natural killer, granulocytes, and whole blood. Today, I will focus on the RNA-seq analysis and the integration with DNA methylation, for which we combine linear modeling and correlation networks. We start with normal and negative binomial linear regression to capture genes with different expression distributions that change with age. So for each cell type and for each gene, we apply both linear models with batch correction and outlier fitting, adjusting for age, sex, and their interaction. To correct for multiple testing, we apply the Benjamini-Holzberg approach and decide on the aging genes based on the beta coefficients of age and their p-values. Focusing on the genes that change linearly with age, we see that adaptive immune cells, see the blue hands, have the largest up-regulated aging effect compared to innate red hands. And that aging leads mostly to upregulation of gene expression across cell types, as we see in the large blue numbers. From these aging genes, we want to see how they share across multiple cell types in up and down regulation. Each color shows the genes that are cell-specific, and the black lines connect the cell types that share genes. We see that aging genes are mostly cell-specific, but we also detect smaller subsets of genes that share aging signature. Next, we want to find genes that co-express within and across cell types, and their expression changes with age. And to answer this question, we used weighted correlation networks. For each cell type, we create a co-expression network based on pairwise correlations on gene expressions by adjusting for age, sex, and their interaction, and correcting for batch effect. We identify modules of highly correlated genes and correlating the eigengene of each module with age, we pick the most interesting module per cell type. And by correlating each module gene to the eigengene, and then to the other module genes, we identify the key drivers in the modules. Thus, we see the most significant module of each cell type and how it correlates with age. And on the right, the distribution of the most significant modules with respect to the intersection with upregulated aging genes in turquoise, the downregulated genes in red, and the ones that are not common with the aging genes in green. While the shared are few, they do follow the same trend of up and down regulation. We see positive correlation matches up with up regulation and negative correlation with down regulation. And although the co-expressed aging gene networks show cell specificity, we do observe shared genes of opposite regulation between cell types. For example, CD8 naives is positively correlated with age, and CD8 effector memory is negatively correlated. And they share genes with opposite regulation. But despite cell specificity, the co-expressed aging genes share across cell types an immunological, senescence, and neurodegenerative disease functional footprint, as I have highlighted in green some examples. Next, we wanted to investigate the epigenetic cis effect of the age-associated gene expression changes, for which we applied a mediation analysis and combined two linear models methylation with AIDS and expression with methylation and AIDS to estimate the indirect effect of DNA methylation on gene expression. Taking the most significant indirect effect indicates that innaids and B-naives have the largest epigenetic effect, which strikingly actually comes from the smallest numbers of mediating methylated sites. Now we wanted to classify these 5K mediating methylated sites by their loci. Comparing horizontally, they are very similarly distributed between gene body and promoter regions, with a little more in favor of down-regulated genes in promoter and up-regulated genes in gene body. We see that the mediating epigenetic effect is also cell-specific, and actually the shared sites are from cell types with the largest number of mediating methylated sites. To sum up, combining linear models and correlation networks, we were able to identify co-expressed aging genes across cell types and reveal the epigenetic role in age-associated gene expression. And our next step is combining them to identify an immune signature with a non-linear autoencoder approach. And with that, thank you for stopping by and hope to see you live. Bye-bye.